CDC proteins. I told you about that 3 that Hartwell did, and also Paul Mars. And then we talked about CDK, cyclin-dependent protein kinases. And I mentioned that of those CDKs, one of them happened to be CDC28, which is the same thing basically. Does anybody have any questions about what we talked about on Friday? Excuse me? What's that? Okay. Okay, so today we're going to talk about, start talking about DNA polymerases and origins of replication, why origins of replication are important. And then Wednesday we'll talk about biochemical identification of origins and assaying origin function. Actually, a couple of things I want to say first, though. I forgot to mention, if you have a Leninger, it's not really our textbook. You can look back at it to remind you of things you heard about in previous years. But I think that the last version of Leninger that I saw, it had a crystal structure in there of CDK2 bound to a cyclin. I don't know if you guys have noticed that. You want to have a look at that. It's a little more detailed than the diagram that I showed you with the 2 blobs on Friday. And also, I can't get a room it's not going to work on the LSI anywhere. So we're going to do for office hours drop-in sessions Mondays and Thursdays at 4 p.m. Be a Zoom session. I'll email the link. The first one will be on this Thursday, so pay attention to that. If you want to drop by and talk about anything, please feel welcome to do that. Not the best, but maybe we can fix this as we go along, but so far I haven't been able to find a place to do this live, unfortunately. Okay, actually last time I was going to ask you, I asked you at the very end, I was telling you about the CDC screen, and I asked, well, why didn't Hartwell get a cyclin gene in his CDC? And somebody said, well, it's because they're redundant, and so the odds are that you'd never get multiple mutations in all the cyclin genes in that particular part of the cell cycle. But I said that there is a possibility that you could get mutations on the cyclins, but they would have to be a special type of mutation. Anybody figured that out? He probably had them, but he probably didn't look at them.
Like a mutation at the site where it binds to the CDK.
Maybe, but genetically, it's a genetic term you need to tell me about here. It's actually one of the first things you hear about in genetics. Exactly, gain of function. They'd have to be dominant, right? So they would have to be mutants in the cyclin genes that somehow had altered structure that it would inhibit the CDK function. And so you probably had them, but dominant mutations are harder to work with, so probably threw them out. So there's that. So today we're going to talk about DNA polymerases. Just to remind you what these things do, this is the substrate for DNA polymerases is single-stranded DNA. This is the template DNA. DNA polymerases, remember, need a primer, and they use that 3' hydroxyl group. The incoming nucleotide, if it forms a base pair with the next nucleotide in the elongating chain, the enzyme catalyzes the use of that 3' hydroxyl group for a nucleophilic attack on the alpha phosphate of the incoming nucleotide, and then you release pyrophosphate. This generates energy for the process, and you end up with the incorporated nucleotide phosphodiester bond formed, leaving a 3' hydroxyl group for the next nucleophilic attack. So that's basically what the enzyme does. You've heard this before. This is just a cartoon of the catalytic site of a typical DNA polymerase. So here's the template strand, single-stranded DNA template, Here's the elongating nascent strand. There's that free 3' hydroxyl group. Here's the incoming nucleotide that forms a base pair. In this case, it's GTP, or deoxy-GTP. It forms a proper base pair with the C on the template strand. And here's the alpha, beta, gamma phosphate. Here's the 3' hydroxyl group forming a nucleophilic attack. So this is typically catalyzed by a couple of charged residues, and there's usually, these enzymes usually have a magnesium ion at the active site to catalyze the formation of the phosphodiester bond. This is from molecular biology, there's a gene book. I've seen this before. And so DNA polymerases all have sort of the same structure. They have this so-called hand structure, cartooned like up there. And so actually it's not drawn properly here. This DNA should be drawn through there. But basically the enzyme is sort of moving this way along the template strand. The hand is moving this way, and then the nucleotides get funneled into the enzyme, and they form base pairs with the template strand, catalyze the nucleophilic attack with the 3' hydroxyl group. Okay, so as I said, molecular biologists like to do this sort of thing where you have a sequence of a gene and you compare it to other genes in the databases, and so it turns out that all DNA polymerases have the same general hand structure, and you can find the fingers. Why is my pointer having trouble? You can find the fingers and the palm and the thumb in sequences. All DNA polymerases that you look at have the same general structure. Most polymerases have additional stuff usually at the N terminus or the C terminus. This adds additional functional properties to the enzyme. We'll talk a little bit about that in a bit here. But basically, the long story short is all DNA polymerases have the same general structure. So, and I think Lineager probably has a crystal structure of a DNA polymerase there as well. Well, I haven't checked that specifically. Okay, so again, you're familiar with this. So bioinformatics, you can take sequences and you have a whole bunch of DNA polymerases and you compare them to each other. You can get an evolutionary tree, looks like. And so if you do that with all the DNA polymerases known to man, they all sort of cluster into 7 distinct families. That's where these families come from for DNA. This is something that's happened over the past 10 years or so. They're now characterizing these polymerases in terms of these families. And so this really has nothing to do with function per se. They just look the same sequence-wise. They cluster into specific families. So there's 7 of them. And so just a couple things I want to point out here is that, so for example, enzymes that you've heard about. So T7 DNA polymerase, actually Taq polymerase, those kind of enzymes, they're in this A class of enzymes. There's a couple of human DNA polymerases in here as well, and also the mitochondrial gamma DNA polymerase is in this A class. So these are all sort of like prokaryotic, simple DNA polymerases. But it turns out that something a little unusual is that the major replication enzymes that we'll talk about, alpha, delta, gamma, or alpha, delta, epsilon, these are actually more closely related to DNA polymerase 2 from E. coli than they are DNA polymerase 3, which you probably heard about a lot in 3rd year. So DNA polymerase 3 in prokaryotes, this is the major replication enzyme in prokaryotes, sort of out by itself in this family organization. Whereas the eukaryotic replication complexes look more like DNA polymerase 2 from E. coli. There are, archaea, you've heard about that, right? They're sort of in between prokaryotes and eukaryotes. They have their own DNA polymerase, not very well characterized. And then there are these X and Y families. So human cells have got polymerases in this family. We'll talk about these When we get to the DNA repair section, these tend to be enzymes involved in DNA repair. And they have some really unusual features that we'll talk about. There's one more polymerase on this family known as the reverse transcriptases, which I'm sure you've heard about. So viruses have these enzymes. These are DNA polymerases, but they use an RNA template instead of a DNA template. And so retroviruses express these things, but human cells also make a reverse transcriptase, and that's telomerase. Telomerase is the enzyme that extends the telomeric repeats at the ends of chromosomes, and we'll talk about how that works. And so these are so if you see in the literature, this polymerase is in the A family, well, you know what that means. It just means it's related sequence-wise to other proteins that are in that family. Nothing to do with function per se. I need to review a few things just to remind you. Some important differences between DNA and RNA polymerases. I've been hung up on this thing the last few years about realize that the more you think about DNA polymerases, they're stupid enzymes. They're not very smart. Because if you compare it to RNA polymerase 2, for example, eukaryotic RNA polymerase 2 is a really sophisticated enzyme, if you think about it, compared to DNA polymerases. DNA polymerases are kind of messy and dumb, I would say. Okay, so why do I say that? Okay, for one thing, DNA polymerases produces DNA product, RNA polymerases produce RNA. Of course, DNA polymerases use DNTPs, where RNA polymerase uses NTPs. Here's the important thing. As I mentioned as well, DNA polymerases cannot initiate de novo. They have to have a primer. Whereas RNA polymerases, they can initiate all on their own. They don't need any kind of primer or anything. So they initiate de novo. DNA polymerases need a single-stranded DNA template. And this requires the extra function of a helicase. So the DNA polymerases have to have a major helicase to unwind the DNA. We're talking about extensive unwinding here. At the replication fork, you get kilobases of DNA unwound, and that's single-stranded DNA. That's the template that the DNA polymerases act on. Whereas RNA polymerase is kind of cool. You can add RNA polymerase plus NTPs to DNA, and it makes its own little bubble, transcription bubble, about 20 nucleotides, and it'll transcribe along. It'll copy DNA into RNA without needing to unwind any DNA. Excuse me. And the other thing about RNA polymerase that's really cool is that it initiates, if you think every gene that RNA polymerase 2 transcribes, it starts at a specific nucleotide. Every transcript starts at that same nucleotide and initiates from that point. Whereas DNA polymerases, they just happen to start synthesis wherever they land. There's nothing special about the start site of DNA polymerases. They just, wherever they bump into the template, that's where they, and if there's a primer on there, that's where they initiate. So there's nothing special about the initiation site, except that initiation happens at origins. But that's just because the replication machinery gets recruited to origins at the start of S phase, which we'll talk a lot about. So no site evidence. Also, the other thing is that DNA polymerases don't have any kind of termination signal. They just go until they bump into something. And so in eukaryotes, you get two replication forks, and how they terminate is they just crash into each other. And that's how they terminate. Whereas RNA polymerase, it has specific signals that tells the enzyme to terminate transcription. RNA polymerase, depending on how you count, minimal 12 subunits. DNA polymerase is the minimal amount of components that you need to get DNA polymerase activity is typically 4 subunits, which we'll talk about a little bit here in a second. One major thing that I'm going to keep coming back to is the fact that the replication field, people studying DNA polymerase activity in vivo, for years and years and years, ignored the fact that replication complexes have to go through chromatin. And I'll come to this in just a second here, but whereas the transcription field, studying RNA polymerase II, has been characterizing this for the last 20 years or so at least. And there's this thing you may have heard about called the C-terminal domain, or the largest subunit. It's like a leash. It carries along with it all of these RNA processing factors, chromatin modifying complexes and things. And so it's pretty well understood how RNA polymerase deals with chromatin. And it's only been the last couple of years or so, I would say, the DNA replication field has even started to consider how it is that DNA polymerases deal with chromatin. And so these enzymes probably carry along with it chromatin modifying complexes. And we'll talk a little bit about that as we get further down in the set of lectures. So DNA polymerases, when they initiate, they just go as fast as they can. As long as they have nucleotides, they'll just keep going. There's no regulation on the speed at which these enzymes travel. Whereas RNA polymerase is kind of cool because it's got these it's got these elaborate systems it's like a gas pedal. It regulates pausing and elongation of the enzyme, and that's important for gene regulation for a variety of instances. Okay, the one thing that DNA polymerases have, at least some of them, that RNA polymerases don't have is an editing function. So you know that some, not all, DNA polymerases have a 3' to 5' exonuclease, and that the function of that is to cleave off a misincorporated nucleotide. If you get a mismatched base, you have a chance to repair that right away just by cleaving off from 3' to 5' exo function. RNA polymerases don't have that. They make a mistake. There's nothing they can do about it really, but they can back up and try again. And so there's this backtracking mechanism that you'll hear about. Any questions or comments on that? You've heard a lot about this stuff before, right? Yeah. Okay, so this is a table that summarizes, this is the list of the known DNA polymerases that are present in a typical eukaryotic cell. There's lots of them. So we have, I don't know how many, there's like 20 of them or so. And so they all have these Greek letters. Alpha, delta, and epsilon are the three we're going to talk about a lot, because these are the enzymes that are our major replication complex enzymes, basically. So alpha, it's known, this is the enzyme that primes DNA synthesis, and delta predominantly, well, exclusively synthesizes the lagging strand, whereas epsilon is the major enzyme that synthesizes the leading strand, but not the only enzyme, because delta can sometimes synthesize the leading strand as well. So these are all, as I said, these are all like from the B family. They sort of look like DNA polymerase II from E. coli. The rest of these guys are mostly for DNA repair, except for where's gamma? Right here. This is the mitochondrial DNA polymerase. This is the one localized to the mitochondria, replicates the mitochondrial chromosome. The rest of them are involved in DNA repair, and that includes these X and Y family enzymes that are involved in various DNA repair mechanisms, except of course for reverse transcriptase. This is the enzyme that does synthesizes telomerase, or the telomeres. Okay, so there's this concept, polymerase switching. And so this is just a cartoon of a replication fork elongating in just one direction. Remember, the replication is always bidirectional, right? So this is just showing one replication fork coming this way. So alpha is the enzyme that primes DNA synthesis on both the leading and the lagging strands. Alpha primes, and there's a reason for that. I'll come to that in a second. And so then, so alpha will, this messy line here, this squiggly thing, that's supposed to represent a bit of RNA. So alpha has on it it's got its own little RNA polymerase stuck to it that synthesizes the first dozen or so nucleotides, but those are RNA nucleotides that are incorporated. And then it switches its own activity from RNA synthesis to DNA synthesis. It makes a bit longer extension, not very long, but goes a bit longer, makes some DNA. And then you get switching of the polymerase to either on the leading strand, primarily you switch to epsilon, and in the lagging strand you switch to delta. And so all DNA synthesis is primed with alpha, and then you get polymerase switching to epsilon and delta after a short synthesis of short primers. Okay, so this is a cartoon again. This is of course not what the structure of these enzymes look like, but they're just stretched out here to show the subunits that make up these three enzymes. Here's alpha, epsilon, and delta. So the blue larger subunit here is the catalytic subunit. So this has got the hand thing. There's the blue thing, that's the hand part of it. The other proteins that are associated with this, these are accessory factors, various other things that modify the function of the polymerase. So alpha thing to note here is that it's got these pri genes or proteins. This is the part of the enzyme that has the RNA polymerase activity. So this part will make initially the RNA primer, and then the DNA polymerase function will take over after a short stretch of RNA synthesis. So that doesn't get very long, because then you switch to either epsilon or delta, depending on whether you're on the leading or the lagging strand. So epsilon and delta, they're very similar enzymes. Epsilon's a little bit more complicated. It's not really clear what all the other parts of epsilon do relative to delta. It's known that epsilon is more sensitive to various signaling events and various physiological stresses and things like that. So it's got more controls built into it, say that. Whereas delta is sort of it's even dumber than epsilon. So, but it does the lagging strand. So we'll talk a lot about this. So you can hear a lot about alpha, delta, and epsilon. We're going to talk a lot about these as we go along. Okay, so there's a few features of polymerases that you need to be aware of. That you probably are, I'm sure you are, you've heard this before, just to review. This is just don't worry about memorizing this table. You just need to know some of the differences that you might expect to find in various polymerases, and that's indicated in this column here. So this just summarizes some features of alpha, delta, epsilon, beta, and gamma. And so these are all nuclear proteins, of course, except for gamma, which is the mitochondrial enzyme. They all have 5' to 3' DNA polymerase activity, not shown on here, but it's inferred. As I said, some of them have the editing function, so the 3' to 5' exonuclease. Alpha doesn't have that, so it can't do proofreading. Whereas the rest of these guys, except for beta, beta is a DNA repair enzyme. We won't talk about it until we get to the end of this series of lectures again. And so the rest of them all have editing function, the 3' to 5' exonuclease. The only one that can prime its own initiation is alpha. It's the only one that's got RNA polymerase activity. The rest of them can't do that. It's an important thing to remember. There is our properties, our features, you probably heard about this, known as processivity and fidelity. What does that mean? So processivity means that it's how many nucleotides it can synthesize before it dissociates from the template. And so alpha is it's got low processivity. It means it doesn't have to go very far before it falls off of the template. Whereas the replication enzymes delta and epsilon, they have really high processivity, so they can synthesize thousands and thousands of nucleotides before they'll dissociate. So that's represented by the processivity. Of course, gamma is quite processive as well. The other parameter to remember about, or to think about, is fidelity. So what is fidelity? It just means accuracy, how accurate the enzyme incorporates nucleotides. And they're all pretty accurate except for beta, but there's a reason for that we'll talk about at the end. And so fidelity is really a parameter of two things. One is, of course, the editing function. So the editing function adds to fidelity of the enzyme, but also intrinsic to the enzyme itself is the natural tendency to make an error in the first place. And so some enzymes are just less likely to incorporate the proper nucleotide than other enzymes. And the DNA repair enzymes tend to have lower fidelity than replication enzymes. And that's just because when you're repairing DNA, you just want to get it done as fast as possible without having to worry about whether you're making too many mistakes or not. And we'll talk about that towards the end. Some of these enzymes are also involved, so most of them are involved in DNA replication. As I said, alpha, delta, epsilon. Beta is not involved in replication. Gamma is required for replication of the mitochondrial genome. Some of them are also, like epsilon, for example, also it contributes to some DNA repair mechanisms that we'll talk about later on. And related to that is the final feature here we'll talk about is called strand displacement, which you may have heard about. This is basically when the enzyme is synthesizing DNA and it runs into double-stranded DNA that's annealed. So the Okazaki fragment, for example, that was synthesized before the one that it's currently doing. So strand displacement simply means when it runs into an annealed or nascent DNA ahead of it, it can displace that off of the DNA. And for Okazaki fragments, that's important because it will run through that Okazaki fragment, which has RNA at the 5' end, right? And so it has to get rid of that somehow. So the DNA polymerase alpha has a good strand. Actually, where's delta? Delta has good strand displacement activity. It's the one that does the Okazaki fragments. It shoves that RNA primer off, and that gets removed by an enzyme called FEN1, which repairs the Okazaki fragments. So alpha doesn't do strand displacement. Epsilon can, but only one nucleotide, which seems like why bother? But anyway, so we'll come back and talk about various features of these and why it's important to think about this, particularly with respect to DNA repair mechanisms and other instances of replication stress and things like that. Any questions about that before we go on? Okay, so here's a figure it's from a really old, ancient textbook. And the reason I'm showing this is because this came from a textbook, and I thought this was amazing because here someone drew a replication fork progressing through chromatin associated DNA template. So this is old, and for decades, no one drew a diagram like this again until last year, if you can believe it. And so this is this issue been telling you about, is that for quite a while, the replication field sort of tended to ignore the fact that these complexes have to replicate through chromatin-embedded template. I'm not sure why it is, actually. The transcription field is maybe 15 years ahead of the replication field with respect to chromatin. But I like this diagram because it shows the nucleosomes. It shows all of the complexes that you're going to hear about. So here's DNA polymerase epsilon, synthesizing the leading strand. Here's polymerase delta, synthesizing the Okazaki fragment on the lagging strand. DNA polymerase alpha is here somewhere. I'm not sure where it is, but here's the helicase that unwinds. We'll talk a lot about this. This is the helicase is a complex known as the CMG complex. We'll talk about what that is, but basically it unwinds DNA ahead of the replication fork. It makes big unwound template, and so of course you know that when the leading strand just can be synthesized continuously. It doesn't have to you don't have to get any dissociation or anything from the template, but the lagging strand, the template has to be looped out, right? Because it's DNA synthesis is anti-parallel. So you loop out the single-stranded template for the lagging strand. This has to be protected from nucleases that are present in the typical cell nuclease or nucleus. So there's a protein called RPA, which is replication protein A. This is the same thing as single-stranded DNA binding protein, essentially. So it simply protects the single-stranded DNA template from degradation by nucleases. And so the Pol alpha synthesizes the primer, Pol delta synthesizes the Okazaki fragments. It does the strand displacement function, displaces the RNA primer from the Okazaki fragment ahead of it. And that's the function of this thing called FEN1 in combination with RNase H. It chops off that RNA flap that's left, and then not shown on here, but you end up with a nick in the Okazaki, between the Okazaki fragments, and that's fixed by DNA ligase. So the Okazaki fragments are processed. And so somehow this whole complex runs through chromatin, and we'll talk, so the last couple of years I've been quite a lot of progress on this, but basically there are these histone chaperones that ride along with the protein and are intrinsic to the polymerases themselves actually. They function to re-establish chromatin behind the replication fork as the complex proceeds. So for example, this is the one I was telling you about. This is a review from 2018. You can see it's got all the stuff I've been telling you about. Here's the MCM complex, the CMG complex, the helicase. Here's Pol epsilon, here's Pol delta, there's a primase. There's this thing called PCNA, which is basically it's like the beta subunit of DNA polymerase III. It's like sliding clamp, enhances processivity. But you notice that what's not on here are nucleosomes. And there's a couple of reviews just came out last year that now people are starting to draw nucleosomes on this again. And so this is just because there's just a gap in knowledge about how these complexes deal with chromatin. Okay, so just to review again as well, you've heard a lot about DNA polymerase III in third year, I'm sure, but basically there are functional equivalents between all of these functions that are involved with the replication fork. There's an equivalent in eukaryotic cells. So as I said, the enzyme that primes in eukaryotic cells is alpha. That's DNA G in E. coli. The major replication enzyme is in E. coli is Pol 3, but that's delta epsilon in eukaryotic cells. There's this enzyme or this protein called PCNA, which just means proliferating cell nuclear antigen. This works like the beta subunit of DNA polymerase III. It's that sliding clamp thing. It encircles the template DNA and the enzyme's stuck to it. And basically it enhances processivity because it doesn't let the enzyme fall off of the template so easily. I talked about replication protein A. This is just the same thing as single-stranded DNA binding protein. It just coats the single-stranded DNA prevents it from being degraded by nucleosomes. There's the polymerase switching is mediated by this factor called replication factor C. This is the thing that it helps load the sliding clamp. It's involved in polymerase switching. Does some other things as well, but basically this is functionally equivalent to another subunit of the E. coli DNA polymerase III. The helicase in eukaryotic cells is MCM CMG complex. This is DNA B, DNA C in E. coli. And then the primers from the Okazaki fragments, you may remember, in E. coli is removed by DNA polymerase I. but that's done using strand displacement and Fen1 in eukaryotic cells. And then to seal the nicks, of course, everybody uses DNA ligase. Okay, so just a brief introduction to DNA polymerases. We're going to come back to these enzymes a lot as we start talking about replication, so don't worry, you just heard a review of what we're going to hear again. We talked about the cell cycle. I told you that we're going to spend a lot of time talking about G1. This is where licensing happens and you form the pre-replicative complex. The origins of replication become competent to initiate. But you don't initiate until you get to the start of S phase, and that simply is controlled by activating cyclin-dependent protein kinase. I talked about how that works. Okay, so we need to talk about replication origins. So origins of replication are important because it sets up a system such that you can ensure that all of the DNA only gets replicated once. So eukaryotic cells have massive amounts of DNA. And so when you initiate S phase, you want to make sure that you only replicate all the DNA only once. And so the way this works is that you limit initiation to specific sites on the chromosome, known as origins. And the reason you have to do this is because once the key event is initiation. Once initiation happens, because DNA polymerases are stupid, they just go until they crash into something, right? So once they initiate, that's it. There's no control over them anymore. And so you have to limit initiation. So the key event is initiation. And so if you limit initiation to origins, the key thing is that then if all you need to do then is ensure that each origin only initiates once in the cell cycle. So the whole business about licensing that we're going to talk about, that's all set up to just ensure that each origin of replication that initiates only initiates once in S phase. That's it. Once it's initiated, that's it. You don't initiate anymore. So that's the key event. That's the key thing about limiting to origins, is that you start from specific places in the chromosome, make sure you only initiate once, and if all that works, then all the DNA is only going to be replicated once. And so we're going to talk a lot about, this is the whole business about licensing. And so in E. coli, you've heard about this before, but basically typical prokaryotic chromosome has an origin of replication. You get an origin binding protein. You initiate replication, you get bidirectional replication, and in a circular chromosome, the replicating forks just bump into each other, and that's where termination happens. But in eukaryotic cells that have linear chromosomes, it's a little more complicated. You have multiple origins of replication on each chromosome, and so each origin has an initiation event. Once they're initiated, it goes. And so in this case, these two replication forks, they'll replicate until they bump into each other, and that's what termination is. And so as long as you make sure that each of these origins only initiates once, you can control to ensure that all the DNA only gets replicated once. Not shown on here, of course, is what happens at the ends, and that's the role of telomerase, which we'll talk about in a different lecture. Any questions about that first? Okay, so we're going to talk today a little bit more about origins, and on Wednesday in a lot more detail, but there are ways of studying origins, detecting them and characterizing them. And so origins of replication in eukaryotes were, and actually most organisms, were originally identified using some kind of a genetic type of a screen. So you can use a functional screen to identify an origin. So just think about how this works is you have a piece of DNA, just a plasmid, so for example, and you throw it into a yeast cell, for example, if it doesn't have an origin of replication, it's not going to replicate, right? So, and so the simplest way to identify origins is to take yeast DNA, chop it up into small pieces, put it into a plasmid, and then throw the plasmid into yeast cells. Plasmids from that's called a library, right? That's a term you're familiar with. So from the library, plasmids that have an origin of replication will be able to replicate on their own. That's the fundamental basis for identifying origins of replication functionally. So this works in things like prokaryotes. You can do it in lower eukaryotes. It doesn't work once you get too high up in the evolutionary scale, it stops working. And there's a reason for that. We'll talk a lot about why that is. But one important thing to note is that the fact that this type of system worked in yeast, I think it moved the field along by about 10 years, I would say, because it allowed the identification of the complex that binds to the origin, which turns out to be the origin recognition complex. So this is a functional screen. You can do it in yeast and lower eukaryotes. It doesn't work so well in human cells. And I'll talk about why that is. You can biochemically detect replication initiation. And we'll talk about this on Wednesday, but there are various gel techniques you can use where you analyze replication intermediates, and you get these bubble and fork intermediates, which you can interpret as being places on the chromosome where DNA replication has initiated. Bubbles and forks, you can sequence short nascent replication products. I'll talk a little bit about that. Use microarrays or next generation sequencing to map places on the chromosome that initiate that you see synthesis the earliest in S phase. We'll talk a lot about that. A couple of years ago, one group showed that if you map the direction of Okazaki fragments, when you see a switch from one direction to the other direction, you can infer the presence of an origin of replication from that. So we'll talk about that as well. There's an indirect way. If you know what protein binds to the origin, so for example, ORC, we know ORC binds there. You can use a technique called ChIP-seq, which you guys have heard about, right? I'll remind you what that is. Basically, you can localize places on the chromosome where the origin binding protein is bound, and that you can infer from that the location of an origin. But this functional screen that I'll tell you a little bit about is really what got this whole field started, because this is exactly what I was telling you about. If you just take a plasmid, so for example, this plasmid has got a wild type copy of this gene for histidine biosynthesis. And if you clone in yeast fragments of DNA into this plasmid library, for example, if you don't have an origin of replication on this, so you transform a strain of yeast that's defective for the same hist gene on the chromosome. And so the only way you can grow, if you don't give these yeast histidine, is if it replicates the plasmid, right? So if it doesn't have its own origin of replication, you get very infrequent colonies that grow, because the only ones that end up growing are ones that somehow the plasmid gets combined into the yeast chromosome, and that occurs at a very low frequency. But if you happen to have a clone that contains an origin of replication, if you take the same yeast strain, transform it into yeast, you get thousands of colonies. And that's because every yeast cell that takes up the plasmid can replicate. It doesn't need histidine anymore because it makes its own histidine. And so this is called high frequency transformation, and it's the assay that was originally used to identify origins of replication from yeast and other or lower eukaryotes. Any questions about that before I go on? Okay, so if you take these yeast that you get these high frequency transformants, take those yeast and extract the DNA from them, you can see that this plasmid, it replicates independently of the chromosome, it replicates as a small circular supercoiled molecule separately from the chromosome altogether. And so if you take those and sequence the inserts, turns out there's a consensus sequence. Don't memorize these kind of things, please. Basically, the yeast origins of replication have a consensus, and this, and so turns out every origin of replication in the genome has at least one, usually more than one, normally 2 or 3 of these ARS, what's called the ARS consensus, which stands for autonomously replicating sequence. It's a consensus element. And so we'll talk on Wednesday about how it was shown biochemically that if you go and you look at on the chromosome where this fragment of yeast DNA came from, and you measure biochemically, is there replication initiating there? And it turns out there is. And so they turned out to be biochemically represent origins of replication. Each origin has at least one of these ARS consensus, usually 2 or more. And then in the yeast chromosome, you get an origin about every 40 kilobases or so across all of the yeast chromosomes. So if you look at this consensus, turns out that there's other yeast species it's sort of concerned. Don't worry about that too much. But basically, as I was saying though, as you get a little higher up in evolution, you lose this consensus sequence. In fact, human cells express proteins that look a lot like the ORC proteins in yeast, but they don't. Human ORC doesn't bind to this consensus. We'll talk about that and what it does bind in a later lecture. But Pombe is weird. It just does a weird thing, so forget about it. Okay, so the important point to take home here is that, as I said, the identification of origins of replication in yeast was an important observation because this sequence was used to biochemically identify what the complex was that bounds to this origin. And this is where the term origin recognition complex comes from. It basically just binds to the ARS consensus element. And so it turns out the OR complex is conserved. All eukaryotes, human cells have got it. Every eukaryote cell has ORC. It's a 6 subunit complex, recognizes the origin of replication, but for some reason, through evolution, ORC lost its ability to bind to a specific DNA sequence. There's something a little more complicated about metazoan ORC, recognition of origins. So we're going to talk about on Wednesday how you would biochemically identify an origin of replication. And this is this so-called Brewer and Fangman 2D gel technique. There is a technique you can use to map precisely nucleotides where replication initiates, but nobody cares about that because, as I said, these enzymes are stupid. They just land where they land. So on Wednesday, we'll talk about biochemical identification of replication origins. So just to remind you again, Thursday, 4 p.m. will be the first drop-in session. I'll post the link